My talk will go into detail on N-glycans, since in my opinion, or of my group, there's an aspect of N-glycans, especially of Fc N-glycans, that is underrepresented in nowadays discussion or even ignored, which is the N-glycan pairing. And I will talk about what in our understanding would be a really comprehensive analysis of this glycan pairing and what this might mean for the impact of N-glycans on the biological activity of our biotherapeutics. So I'll start with an introduction on N-glycans, Fc N-glycans in general, to bring us all on the same page. In general, N-glycosylation is a very common, usual post-translational modifications of eukaryotic proteins. It's used by biology to label proteins to influence cell-cell interactions, protein-protein interactions, protein receptor interactions, whatever. And the majority of secreted eukaryotic proteins carry N-glycans. And so do our monoclonal antibodies, especially in the Fc part. There's a conserved region at asparagine 297. And all of the Fc-containing proteins are glycosylated in this place. It's usually depicted like this in the pictogram, but it's more accurately to look at the structure, where you can see that the N-glycans are actually buried in the Fc and help to form this horseshoe conformation of the Fc, which is also responsible for Fc receptor binding and so on. There are many different types of glycans. In terms of Fc N-glycans, it's mainly these three, which are separated into high-mannose because they have exclusively mannose structures after the core structure. Complex N-glycans, which are characterized by N-acetylglucosamine and a varying degree of galactose residues, sometimes sialic acid, but that's rather not that much abundant in Fc N-glycans, and the hybrid type, which is just a combination of the aforementioned. Due to the multitude of possible linkages and sugar moieties in N-glycans, we have a high degree of heterogeneity in general. And unfortunately, these types of glycans, this heterogeneity, influences some of the important functions of Fc-containing biotherapeutics. For example, the stability of the Fc part is influenced by the N-glycans, pharmacokinetic properties, pharmacodynamic properties, the effector functions of the Fc are mainly influenced by the N-glycans. To go a little bit more into detail on that, so from generally speaking to more details, as I said, the conformational stability of the Fc is maintained by the N-glycans. If they're missing, the Fc has different conformation, different properties. This type of glycan, the high mannose, in this case mannose 5, because it's 5 mannose residues, is kind of important, and I will focus on these type of glycans in the rest of my talk because they're associated with accelerated clearance in the body. So mannose-containing, high mannose glycan-containing molecules are cleared faster from the bloodstream than other types of glycans. Another important example from the complex type glycans, which everybody probably knows, is the core fucosylation, which influences the binding to Fc gamma 3A receptor, which then influences the antibody-dependent cellular cytotoxicity. Galactosylation is also an important factor which can influence the binding to the 1Q compartment of the complement system, thereby influencing CDC, or further interactions with other Fc gamma receptors is also influenced by galactosylation. Now that was basically very general knowledge about N-glycans, everybody should know, and this is now where it's getting interesting. As already mentioned, the Fc part contains 2 Fc N-glycans, which we more accurately refer to as glycopair, because it's obviously a pair. But there's symmetrical as well as asymmetrical combinations possible. So we do not necessarily have the same type of glycan on both chains on both heavy chains in our Fc part. There could also be combinations of different glycans. But when we analyze glycans, we heard of that yesterday and today a lot, we almost exclusively employ release glycan assays, glycopeptide assays, or subunit assays where we actually miss the information on the actual distribution of the glycans we measure absolutely on the proteins. So how are they actually distributed? Because, as I told you, asymmetrical combinations are possible. If you look at this example, this may highlight why this might be important. So if we measure an absolute amount of 20% mannose-5 in our drug product, drug substance, there's two possibilities. If we assume exclusively symmetrically pairing, so there's on one antibody molecule, two M5 glycans, we have 20% of our molecules carrying mannose-5. But if we assume asymmetrically pairing, there is 40% of our molecules carrying at least, or in this case, exclusively one mannose-5 glycan. So there's a big difference in how we can assess the impact of these type of glycans if we don't know how they're actually distributed over our molecules. And I will show you later that the pairing of these high mannose molecules is important. It does influence binding to the mannose receptor, which is meant to be responsible for the accelerated clearance in vivo. And furthermore, other combinations of asymmetrical paired complex type glycans are largely unknown. So no one ever looked at that in particular. So as I told you, release glycan and all these methods, we miss the information on the apparent distribution. So we need to leave the Fc part intact to get the information on the pairing. So we need to employ intact mass. So this is just a regular spectrum acquired with BioAccord system. All the data I will show are acquired easily by a BioAccord system with a simple method. I don't have a picture of it in the presentation, but you might have seen it a lot these days. You can see there are asymmetrical pairs. The usual distribution would be the G0F, 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 G1F. So there are some asymmetrical pairs everybody is aware of. But where is the rest? So from a statistical point of view, there should be more asymmetrical pairs. For example, also mannose-5 asymmetrical pairs. So the intact spectrum alone isn't really sufficient to really get the information on the actual distribution I was talking about, because the spectra tend to be busy, overlapping, there's isobaric species, so we need another approach. Which is why we tested a series of different glycosidase enzymes to reduce spectrum heterogeneity, which resulted in this. So we came up with the, we ended up with endoglycosidase F3 as the enzyme of choice because it specifically cleaves off fucosylated complex type glycans. And what we are left with in the spectrum is 3 dominant peaks. So this is the same IgG enriched in mannose-5 to increase sensitivity and visibility of the species we are looking for. So we cleave off all the fucosylated complex type glycans and are left with the undigested mannose-5 pair, all the digested complex type glycans, but in between we have this asymmetrical pair. So we could show they are present in the sample and they are quite high abundant. When we then calculate the intensities and the relative abundances of the 3 peaks, we come up with the number of 33% of the mannose carrying antibodies in the sample are actually asymmetrically paired, which translates into a 20% underestimation of the number of mannose-5 carrying molecules. That is that fits to the example I showed in the beginning. So we underestimate the actual number of molecules carrying the mannose-5 in this case. Now that we can quantitate it and identify that there are asymmetrical glycopairs, we were interested in what are the pairing partners. So what was on this side? Does this give us any hint on the glycan processing in the cell or further information? So we just reversed the approach and cut off the mannose-5 glycans with ENDO-H, which is really specific for mannose-containing glycans and leaves the complex type glycans intact. So we again have the digested former mannose 5 peak, but we can identify here all the pairing species which were summarized in the one peak in the spectra I showed before. And we can see that there are a lot of kind of truncated, incompletely processed glycans which pair with the M5, which might be a hint on incompletely or inefficient glycan processing. If there's a mannose 5 on the one side of the Fc, that the other side might not be glycosylated that efficiently. And what we also found, just as a side note, the undigested part of the spectrum is also showing a lot of truncated complex type glycans, which are also species we didn't really see before, and which also confirms the specificity of our mannose enrichment process. So all the terminal mannose residues show that the enrichment was really specific for the terminal mannoses. So now that we learned how to quantitate and identify the asymmetrical pairs, we did a larger study on different types of molecules to see really, is this molecule specific, where is this coming from, is this dependent on the format. So we calculated the overall mannose 5 content of all the different molecules, and also how is the distribution of asymmetrical pairs and what are the pairing partners. This included many different formats, from IgG1, IgG4, 2 different bispecific formats, Fc fusion proteins. These were from different cell lines, CHO as well as SP0O, which is murine, I think. They had different Fc backbones, so there were LALA mutations, or the IgG4 had these SP mutation for the prevention of Fab arm exchange. So many different molecules, different scaffolds, formats, backbones, different cell lines, and we could conclude that we can't conclude anything. We didn't find a clear correlation between the distribution of the asymmetrical pairs and the molecular properties. So this made us conclude that this is a very, very specific property of the molecules, which has to be assessed on an individual basis. So I have my molecule, I can't just estimate from the cell culture process or from the format that the distribution of my glycans are measured in absolute amounts can easily be calculated. You'd have to assess this on an individual basis. I have to mention that I think this is not enough data to really employ statistical approaches. We would need more sophisticated choice of molecules and then make maybe a statistical analysis of this phenomenon, but we are pretty sure that this is really individual phenomenon. What we also had a look at was the process. So we just choose an example process which is known for high mannose content in the end, and measured the absolute M5 glycan amount and the distribution of asymmetrical pairs. So as you can see from the black curve, by the end of the process, the amount of high mannose in the process goes up. This is not really surprising. We have cellular stress in the end. A lot of product, mannose 5 will increase because of incomplete processing. With the green bars, you see the absolute amount of M5 carrying molecules, which is also going up. Also not surprising, we have more M5, so there's more M5 carrying molecules. What is more surprising is the blue bars, which is the portion of the asymmetrical paired high mannose in the sample, which is going down by the end of the process. So again, this is relative, but it's going down. We might have expected that by the end of the process, with an increased amount of mannose 5, we would also have more asymmetrical glycopairs due to this being a product of stress, but it's actually not the case. The asymmetrical pairs are formed throughout the whole process and are rather going down by the end of the process. So we can say that it's not exclusively product of cellular stress, but it happens anyway during the whole process. Okay, now that I've talked a lot about how to analyze it and what we found, I try to convince you that this is actually important and relevant for biotherapeutics. This is part of the work of my esteemed colleague. She's also a PhD student and working on the mannose receptor map interaction. Therefore, she built an affinity column with immobilized mannose receptor and did affinity chromatography of different high mannose containing molecules to study the interaction. You can see here a chromatogram, and what we find is that the elution fraction is exclusively symmetrically paired high mannose. So this high mannose does interact with the mannose receptor, binds pretty strong, and is retained on the column. Not surprising, but when we look at the flow-through fraction, this was fractionated and analyzed with the presented methods for the glycan pairing. We find in the beginning of the peak mostly complex type glycans, symmetrical complex type glycans, but in the end of the peak, the tailing, we have accumulated asymmetrical high mannose containing glycopairs. So the asymmetrical pairs with one mannose are not really retained, bound by the mannose receptor as the symmetrical pair, but they do actually interact with it. So they're a little bit retained, but just a little bit of tailing. But in conclusion, means that the symmetrical pair, double mannose, does behave differently than the asymmetrical pair, which is why it has to be assessed with the pairing information if we want to know how critical is our mannose content for example, for the clearance and therefore for the pK of our molecule. So now I've talked a lot about mannose, but there's for sure other glycans. Just to give you a little outlook on what is currently going on. We're also looking at asymmetrical complex type glycans for sure. And this just as an example why this is also important for this type of glycans. This is just the endo-3 digest of a usual IgG with very low content of R-fucosylated glycans because it's a silenced molecule. We don't want the effector functionalities in this case. And as you can see from the zoom here, the bigger part of the R-fucosylated N glycan is actually asymmetrically paired. So again, if we have a silenced molecule, for example, and have an amount of R-fucosylated glycans, we actually need to know how are these glycans distributed over the molecules to really assess the criticality if we don't want the effector functionalities and so on. And as you can see, most of them are asymmetrically paired. Now we have to think about or assess, is this critical? How do these asymmetrical pairs actually interact with the Fc receptors? And what is the actual amount of our molecules carrying these R-fucosylation? This is part of current work. Stay tuned for that. I'm already at the end of my talk. I just want to briefly summarize what I was talking about today. We presented methods to assess the actual distribution of N glycans over the molecules with our specific glycosidase digest, with the example of mannose-containing glycans. We tested these methods on a whole set of different molecules and assessed the asymmetrical glycan distribution and found out to be really individual, which means it needs to be assessed on the individual basis and can not just be estimated. It is important, as shown by the preliminary results from my colleague. So we talked about the clearance of the high mannose-containing species. We talked about R-fucosylated and the impact on Fc receptor binding and, for example, ADCC, and which is part of ongoing work. We want to evaluate further effector functions, further asymmetrical glycopairs. How do asymmetrical fucosylated pairs affect Fc receptor binding? How do asymmetrical galactosylated pairs affect CDC binding? And what would it mean for biological functions? Therefore, we employ SPR analysis, and in the future, it is planned to confirm the results in real biological assays to really make the conclusion from our glycan analysis, glycopairing analysis, up to biological function of our therapeutics. That being said, I also want to thank my team at Boehringer Ingelheim, Julia Baumeister, my co-PhD student for her results, my supervisor, and my doctor father at Ulm University. These are my references. Thank you for your attention, and I'm happy to take any questions.